Today we explore how artificial intelligence can personalize nutrition while respecting genuine biological mechanisms, not diet fads. Artificial intelligence connects individual biological data points to create personalized nutritional guidance, treating each person as a unique metabolic system. Why do two people eating identical meals show drastically different metabolic responses? Person A experiences a sharp glucose spike after eating bread, while person B shows a gentle curve. This is not preference or psychology, this is biology. 4 major biological systems drive individual nutritional responses: glucose regulation, gut microbiome composition, sleep architecture, and physical activity patterns. These 4 pillars interact continuously. Your microbiome affects glucose handling, sleep influences insulin sensitivity, and activity modulates all 3. Let us begin with glucose dynamics, the most measurable metabolic output. 3 people consume 50 grams of white bread. Continuous glucose monitors reveal 3 distinct response curves: sharp spike with rapid decay, moderate peak with slow return, and intermediate dynamics. Question: what biological mechanisms create this threefold variation in glucose response to identical carbohydrate loads? Answer: 4 primary factors: insulin secretion kinetics, skeletal muscle glucose transporter density, incretin hormone profiles, and gastric emptying rates all vary by individual genetics and physiology. We can model glucose response as baseline glucose plus a Gaussian peak, characterized by amplitude A, time offset tau, and width sigma, all person-specific parameters. Insulin secretion from pancreatic beta cells triggers glucose uptake. The magnitude and timing of this cascade differs dramatically between individuals due to receptor sensitivity and transporter expression. The gut microbiome, a community of trillions of microorganisms, profoundly influences nutrient metabolism and energy harvest. A simplified representation of gut microbiome diversity. Yellow circles represent firmicutes, blue Bacteroidetes, pink other phyla. Each person hosts a unique ecosystem. The ratio of firmicutes to Bacteroidetes, 2 dominant bacterial phyla, correlates with energy harvest efficiency from food. Higher ratios extract more calories from identical meals. Energy harvest can be modeled as alpha times the firmicutes to bacteroidata ratio, plus a baseline beta. Alpha and beta are empirically determined constants. Gut bacteria ferment dietary fiber into short-chain fatty acids, acetate, propionate, and butyrate. These metabolites regulate appetite, inflammation, and insulin sensitivity. Question: how much does microbiome composition actually affect caloric absorption from a standard meal? Answer: studies suggest microbiome differences can alter daily energy extraction by 50 to 150 kilocalories for identical diets, accumulating to significant weight changes over months. 5 individuals consuming 2,000 kilocalorie diets show absorbed energy ranging from 1,900 to 2,100 kilocalories due purely to microbiome differences. Sleep is not metabolically neutral. Sleep deprivation acutely impairs glucose tolerance and insulin sensitivity. Sleep architecture alternates between REM and deep sleep stages. Deep sleep, particularly slow-wave sleep, is critical for glucose metabolism and hormonal regulation. Insulin sensitivity IS decreases linearly with sleep debt delta T, where K is approximately 0.1 per hour of lost sleep based on metabolic chamber studies. Identical meals consumed after 4 hours versus 8 hours of sleep produce dramatically different glucose curves. Sleep deprivation amplifies postprandial glycemic excursions. Sleep loss elevates cortisol and activates the sympathetic nervous system, both inducing insulin resistance and hepatic glucose production. Physical activity acutely and chronically modulates glucose metabolism through multiple pathways. Muscle contraction triggers GLUT4 glucose transporter translocation to the cell membrane, enabling insulin-independent glucose uptake that persists for hours post-exercise. Glucose uptake equals basal uptake plus beta times metabolic equivalence times duration, where beta reflects individual oxidative capacity. Exercise redirects substrate flux. Muscles increase glucose uptake, while adipose tissue mobilizes fatty acids, sparing glucose, and improving whole-body insulin sensitivity. Let us calculate. How much does 30 minutes of moderate exercise affect glucose disposal? 30 minutes at 5 METs, roughly brisk walking, consumes approximately 750 kilocalories, predominantly from glucose and fatty acids. If half comes from glucose at 4 kilocalories per gram, that is 94 grams of glucose cleared, substantially lowering postprandial glycemia. Now we integrate these four pillars into a computational framework. How does I synthesize this complexity? The AI model ingests four continuous data streams: continuous glucose monitoring, microbiome sequencing, sleep tracker data, and activity logs, fusing them into personalized predictions. The predicted glucose trajectory Y at time t is a function of food composition, microbiome state, sleep quality, recent activity, and individual parameters, theta I. Question: what machine learning architectures effectively capture these non-linear, time-dependent interactions? Answer: ensemble methods like random forests and gradient boosting handle tabular features well. Deep neural networks, particularly recurrent architectures, excel at temporal patterns. Predicting personalized glucose response resembles weather forecasting. Both are chaotic dynamical systems with measurable initial conditions. Weather forecasts integrate atmospheric pressure, humidity, and temperature to predict future states. Glucose forecasts integrate meal composition, microbiome, and activity to predict metabolic states. Both weather and glucose dynamics exhibit sensitive dependence on initial conditions. Small measurement errors amplify over prediction horizons. Prediction uncertainty sigma grows exponentially with time constant lambda, the Lyapunov exponent, limiting forecast horizons to hours or days. What volume of data enables accurate personalized predictions? Continuous glucose monitors provide 288 measurements daily. Combined with meal logs, sleep sessions, activity streams, and periodic microbiome samples, we generate thousands of data points weekly. We need at least 14 days of continuous data to begin building accurate, personalized models for glucose prediction. To summarize, we have discussed the four pillars of biological variability that influence nutritional responses: glucose regulation, gut microbiome composition, sleep architecture, and physical activity patterns. By leveraging machine learning algorithms and integrating data from these four pillars, we can build predictive models of glucose response that enable personalized nutrition and health recommendations.